All right, the next presentation here is from Shuttle Pharmaceuticals Holdings. Presenting from the company today is Dr. Anatoly Druchillo. He's the company's chairman and chief scientific officer. Tony, take it away.
Thank you very much, Robert. Good afternoon, ladies and gentlemen. I'd like to update you. I did this presentation about a year ago, and I'd like to update you on some of the things that have happened, some of the milestones we have reached. This is the forward-looking statement slide, which you're welcome to read on our website. And what I'd like to do is to address the fundamental problems in radiation oncology as I see them. Basically, the technology to improve radiation oncology has all been focused on machines because these are multi-million-dollar machines, and they're basically designed to treat smaller and smaller volumes of tissue. However, we can give enough radiation therapy to put a hole in the patient so that we have to be careful, and we have to basically try and look at the science underlying the failures of treatment. And the science relates to the fact that many cancer cells are let's say as resistant to radiation or as sensitive to radiation as are the surrounding tissues. And some cancer cells are even more resistant. So the only way you can actually improve the treatment beyond what is currently done with beam shaping is to affect the biology. And we have two approaches to it. One is the sensitization of the cancer cells to radiation therapy. And we are currently in the phase 2 clinical trial of a drug that we propose to use for this. And also, we have the ability to stimulate the immune system after radiation, which is activated by damage to cells by the radiation, and to end up with an improvement in overall survival by treating the patient post-radiation therapy. Today, I'll also touch on some of the new technology we've developed within the company, new intellectual property, which includes the theranostics for both diagnostic and therapeutic applications for prostate cancer. The overall company structure is shown in this slide. Shuttle Pharmaceuticals Holdings is a holding company under which two companies function, Shuttle Pharmaceuticals and Shuttle Diagnostics. They are both wholly owned, and we're looking at options for enhancing the ability of Shuttle Diagnostics to raise capital in order to develop some of the technologies to clinical applications. The therapeutics, we have ropedoxuridine as a clinical stage drug in phase 2. We have SP225, which is the preclinical stage lead selective HDAC6 inhibitor. The diagnostics, we have a prognostic test, a PC-RAD test, where a blood test is obtained prior to radiation therapy to determine the patient's response to radiation therapy. And the other is the PCMA, prostate-specific membrane antigen ligand, which is a theranostic molecule that we have recently invented. The mechanism I've previously reviewed, but basically we have a prodrug called IPDR, which is metabolized to the active ingredient. The active ingredient gets into the DNA. By being in the DNA, it makes the DNA more sensitive to radiation injury, and you have cell killing. Our phase 2 clinical trial is approximately halfway through the initial part, the randomized part. We were planning to compare this to historical controls. The patients that we selected are the most aggressive glioblastoma patients that there are. These are methylation negative IDH wild type. They do not respond to temozolomide, the drug that is frequently used in treating brain tumors in combination with radiation. And so they constitute about half the patients that present. So we're treating a group of patients for whom standard of care is radiation only. And by adding the drug, we wish to determine if we can prolong the survival of these patients. Typically, these patients die within a year of diagnosis. We'd like to improve that by going from, let's say, a 40 or 50% survival at 1 year to a 75% survival to show that there is efficacy of the drug. Patients are randomized to 1,200 milligrams per day plus radiation versus 960 milligrams per day plus radiation. At the end of this, dose levels and the complications or side effects will be compared, and the optimal dose will be determined for an additional 14 patients to get to the current number of patients that are required for statistical significance of 34. Okay, we also have about 20 patents on selective HDAC inhibitor compositions of matter, and we have identified two of them as our candidate leads. One is the SP225, which activates the immune response post radiation treatment, and we would like to develop this as we go forward when there is available funds, since much of it is not currently engaged in promoting our clinical trial. The other one is SP1303, which inhibits estrogen receptor positive breast cancer cell growth. And it is a molecule that we would like to develop in conjunction with another group that we're currently speaking to. The clinical and preclinical development strategies are listed on this slide. I'm not going to go through each of these. The slides will be on our website. However, the primary goal, primary mission is to complete the development of ropidoxyuridine plus radiation therapy. The second goal would be the 225, the selective HDAC6 inhibitor and immune modulator. And the third would be the inhibitor of breast cancer cells with estrogen receptor on them. Over the past several years, we have also been fortunate to receive government funds, contracts to develop diagnostic tests for radiation-related indications. This is the PC-RAD test that we developed while I was still at Georgetown University. This has been licensed by Shuttle, and all of the testing in terms of the analytical validation have been completed. We now are prepared to do a clinical trial, which we will be looking for funding for this in trying to develop it as a diagnostic test for prostate cancer. These are the various important, let's say, considerations of why we should do this. The main one is that a patient who's going to undergo radiation, if they can be predicted to either do well or poorly, can be an important, let's say, assistance to both the patient's decision-making and the physicians. The second area is a little more complicated. This is the PSMA ligand. The PSMA ligand is on the surface of prostate cancer cells. It's an enzyme and it's an antigen, so that the body does recognize this area. So what happens is a ligand then is like the key that fits into this keyhole and it attaches. The ligand subsequently gets internalized into the cell. And if you attach a radioactive source to this ligand and it gets in and gives off the radiation, you can do PET scanning with it. And Lantheus has done that with a molecule that was developed through Johns Hopkins. And the other one is if you put a heavy-duty radiation source, maybe a little more powerful, more killing, DNA damaging source, then you can have a therapeutic, which is called a radiopharmaceutical. And Novartis has developed Pluvicto and is marketing that now. We have a new ligand that was developed by Professor Alan Kozakowski, who is actually, by coincidence or a chance, was also the person that developed the original PSMA ligand on which all of the other molecules are based, including the current ones that are in the market. So we have the ability to use a very highly selective ligand in order to advance certain aspects of therapy, including radiopharmaceuticals and also, let's say, delivering other drugs using this as a selective pathway. This is an example, and what I mentioned just previously. On the right is a PET scanning machine, and this below it is a scan of a patient showing you the multiple areas of involvement. The dark spots are take up by a prostate cancer that is spread in this patient. So we have that intellectual property within the company, and we're looking to advance it further with preclinical studies. The diagnostics, so we will be developing the PC-RAD test and developing the PSMA ligand. And our plans are described in the slide, and we will be reporting this as we go along, as we go forward. The Shuttle Pharmaceuticals Holdings, in terms of a summary of the intellectual property we have of the ropidux-uridine, we have technology for manufacturing and for method of use patent in combination with formulation with tipiracil to prolong its biological half-life. The selective HDAC inhibitors, we have over 20 patents, U.S. and international. That again, they're in our SEC disclosures, and then of course the predictive PC-RAD test, which we have an exclusive license from Georgetown University, and the PSMA-B, which we have an exclusive license, was developed in combination with our research team. This is the financial overview of the company on March 13th, provided by our CFO, and we have a total of 30 million shares outstanding. The stock price has been suppressed for many reasons, which I won't go into. However, we're looking to, we did a recent closing, and we'll be looking for additional capital raise. The leadership team remains the same, with the exception of the addition of Christopher Cooper, who has taken over the CEO responsibilities, and which include capital raise. And I've continued with the scientific and the chairman of the board responsibilities. Since the last visit, the other people have been stable. Tim Lorber has joined us as an experienced CPA and Chief Financial Officer. That's about all that I have for you. And are there any questions?
Yeah, here, I'll go ahead and start off, and then we'll open up to the room here. Let's come back to the clinical trial, phase 2 clinical trial for ropidux-uridine. I think you mentioned you're about halfway through on enrollment. Talk about sort of expected timelines to complete the initial 40 patients there.
Right. So we've entered the patients into the randomized part, and we have a total of 6 cancer centers on the East Coast that have been engaged in this. We have the ability to expand our enrollment rate because 3 of them have enrolled most of the patients. The other ones have been completing bureaucratic items, but they're all now approved. The IRBs, institutional review boards, have approved the protocol, and we're accruing now a rate of 4 per month. And I expect that may go up, but again, it's not predictable. However, 4 per month has been our track record over the past 4 or 5 months. So I think that we're looking at about between 6 to 8 months, as when we complete the enrollment in the first part, and then another 3 to 4 months for the 14 patients that would be necessary to complete the entire clinical trial.
Let's talk about those 14 patients there, right? And it's talked about at an optimal dose. How are you sort of determining, or what's going to be the factors that go into determining the optimal dose there?
Right. So the optimal dose was something that the FDA is very much interested in, and the design of clinical trial took their interest in into that, obviously, so that we had to establish two different doses. In this case, the dose at which we saw toxicity in patients was 1,500 milligrams per day over a period of three weeks, and the major dose-limiting toxicity was diarrhea and dehydration. So at 1,200 milligrams, we did not see any toxicity. So in a way that, I'm not expecting much toxicity. But then we took this lower dose, which is 960 milligrams, and we'll be testing that. If the two arms are the same, and the toxicities are, let's say, more or less in one or the other arm, that'll help guide us into which dose we take. Right now, it looks like both will be comparable in terms of toxicities, and we'll just have to finish out the randomization and the clinical trial, so we'll be able to test that. The blood levels of the drug are also obtained, and those blood levels have to be in a certain range. And if they're in a certain range, if for one arm or the other, that'll also help us determine which arm we use. I expect that both of these will have some effect, but certainly the 1,200 milligrams is not a problem for us.
Maybe somewhat premature, but talk about how the design of the phase 2 and what the potential outcomes would be might determine what a phase 3 trial would look like, size, patient size, how the study design may go.
Right.
Anything that you might be able to derive from your interactions with the FDA to this point?
Right. So finishing this clinical trial, if we see a positive effect in terms of the survival, and we have a survival endpoint at 1 year, if that is, an endpoint that we reach, then this becomes fairly straightforward and easy to do, because the standard of care is radiation only. And we would take our optimal drug dose and we would add it and compare to standard of care in a randomized trial. And there would just be the necessary numbers for obtaining statistically significant results. Typically, a phase 2 trial will include 100 or more patients or several hundred patients. But because this is an orphan disease and because there's nothing else available, we would also seek breakthrough, try and obtain breakthrough approval from the FDA. And at that point, we can market the drug even while we're doing a phase 3 clinical trial.
All right, that's helpful. One more question on my end before if anyone else has any. Talk a little bit about the diagnostics subsidiary. What are some of the plans to sort of develop that additional intellectual property that the company possesses there?
Right. So the structure of Shuttle Pharmaceuticals, as I showed you before, is that it's a holding company with two subsidiaries. However, one or the other, or eventually, I guess both could be partially spun out for additional, let's say, capital investment and enable us to proceed. We would spin it out with part ownership of by the holding company, so that any investors would benefit from any success we would see with the spin out. But that's all something that we have to pay some lawyers some fees for them to work that out for us.
Understood. Any questions from the audience here by chance?
Yes. So we're in the position where, because of the limitations of how much we can raise at any given time with this post IPO, we'll be raising every, let's say, 6 to 8 months. So right now we have a good cash position, but we're planning to do an additional raise. So if you have any money, I'd like to talk to you. I'd like to think that, but I'm going to tell you something. In my experience, every time I ask for 2 million, the people give me 1 million. Every time I ask for 4 million, they raise 2 million. No one believes what I say. Okay, but the numbers that I give are usually based on good science. There must be a reason that it's always shortchanged.
Any additional questions?
All right,
All right, all set. Thank you very
Thank-
much, Dr. Chillo.
thank you.